Welcome back. Today we're diving into something that sounds like it's straight out of a sci-fi movie. How do you literally pause life? I mean, think about it. Hitting the pause button on biology. But, if you just tossed a living cell into your freezer at home, you wouldn't get suspended animation, right? You just get, well, a dead cell. So how do they actually do it? What's the secret sauce?
Just take a look at this. What you're seeing is a place where time, for all intents and purposes, just stops. Scientists have cracked the code on cooling living cells to temperatures so low that all biological activity just freezes in place. And today, we're going to unpack exactly how they pull off this incredible feat.
Okay, but before we jump into the deep freeze, we've got to take a step back. We really need to understand why it's so ridiculously hard just to keep cells alive outside of the body to begin with. Seriously, that's a huge challenge all by itself. So the whole game starts with this thing called animal cell culture. It's the foundation for almost all modern biological research. The basic idea is you're creating a little five-star hotel for cells in a lab dish. You give them this special liquid food, this artificial medium, to keep them happy, alive, and growing. And here's the thing, cells are incredibly high maintenance. For starters, they grow so slowly. We're talking 12 to 28 hours just for them to double. They're super fragile, really sensitive, and most of them are kind of clingy. They need a surface to attach to. But the real kicker, the biggest problem of all, they're literally programmed to die. Yeah, that's the heart of the problem, right there. Cells have this built-in self-destruct sequence called apoptosis. It's like a little timer in their DNA that's set to go off. So the scientists, they aren't just playing caretaker, feeding them and keeping them warm. They're in a constant battle against the cell's own internal mission to self-destruct. Okay, so let's say you do it. You beat the odds, your cells are happy and growing. Awesome, right? Well, not so fast. Your success actually creates a brand new headache. You've just signed yourself up for an endless, super demanding cycle of upkeep.
The best way to think about it is like a city that's run out of space. Cells grow in this single layer, and once they've covered every inch of their dish, it's total gridlock. They get overcrowded and just stop growing. There's a name for this, confluency. And when a scientist sees that, it's a huge red flag that they need to do something and do it quickly.
And this is exactly what it looks like under the microscope. See on the left? Plenty of room to grow. But on the right? That's a completely full dish. That's what we call a confluent monolayer. They've officially run out of real estate, and the clock is ticking for the scientist. So what do? The solution is a process called subculturing. They basically use a special enzyme to dissolve the glue that holds the cells to the bottom of the dish. Then they scoop them up, thin them out, and move a small batch to a brand new dish with fresh food. It's almost exactly like repotting a plant that's outgrown its pot. The only catch is you have to do it over and over and over again. And you can imagine, right? This endless cycle is not only expensive and takes a ton of time, but every single time you do it, you risk contamination or weird genetic changes. It's a huge problem. Researchers were desperate for a better way. They needed to find a way to just hit pause. And that's where cryopreservation comes in. That's the fancy scientific name for what is essentially the biological pause button. The idea is simple. You cool the cells down to incredibly low temperatures, and every single biological process, all the chemical reactions, just grinds to a complete stop. They're preserved in this perfect state of suspended animation. And when I say low temperatures, I'm not kidding. We're talking seriously cold. The gold standard here is liquid nitrogen. It keeps the cells at a mind-boggling minus 196 degrees Celsius. I mean, at that temperature, time just ceases to exist for these cells. But here's the big plot twist. This beautiful, elegant solution has a massive, deadly flaw. It turns out the very act of freezing, the thing that's supposed to save the cells, can actually destroy them. So what's the problem? Well, it all comes down to one thing. The number one enemy in this whole story is ice. It's the formation of these sharp, jagged ice crystals that can just shred a cell to pieces. And ice has a bunch of different ways to kill a cell. First, as the water freezes, all the stuff dissolved in it gets left behind, turning the remaining liquid into this toxic, super concentrated goo. Then you've got ice forming outside the cell, which can literally crush it. And as that outside ice forms, it sucks water out of the cell, causing it to dehydrate and shrivel up. But the absolutely deadliest thing, That's when ice crystals form inside the cell. That is almost always game over. So the big question is, how do you possibly defeat an enemy like ice? Well, over decades of work, scientists came up with two really brilliant strategies. They figured out how to not just freeze cells, but how to actually cheat ice. Okay, so the two main techniques are slow freezing and vitrification. With slow freezing, the goal isn't actually to stop ice from forming, it's to control where it forms. By cooling things down really slowly, about 1 degree a minute, you give the water inside the cell time to escape. That way the ice only forms outside the cell where it's way less dangerous. Now, vitrification is the more radical approach. The goal there is to prevent any ice from forming at all. You do this with super fast cooling and a cocktail of cryoprotectants. Think of it like cellular antifreeze, which turns the inside of the cell into a solid, glass-like state. No crystals, period.
And what's just wild is the difference in speed between these two methods. I mean, look at these numbers. Slow freezing is trucking along at maybe 0.3 degrees Celsius per minute. Vitrification, it's a mind-blowing 20,000 degrees Celsius per minute. It's like the difference between slowly walking down a hill and being shot out of a cannon into a solid, glassy state.
All right, so you've done it. You've paused time, the cells are locked away in this deep, frozen sleep. But now, how do you wake them up without destroying them? And more importantly, why do we even go to all this trouble? Why does it matter?
Well, it's all about speed again, but this time in reverse. If freezing is slow and careful, thawing has to be the complete opposite. It has to be crazy fast. You literally take a frozen vial and plunge it into a warm water bath, and you want it thawed in less than 60 seconds. This lightning-fast thaw is crucial because it stops any tiny, dangerous ice crystals from reforming. Plus, it quickly dilutes those antifreeze chemicals, which can actually be toxic once they're not frozen solid. And this is why it all matters. This science is an absolute game changer. It means scientists can create these safety deposit boxes of precious cells, which saves a ton of time and money. It's basically an insurance policy against disasters like contamination. It also gives them a reliable, consistent baseline for all their experiments. And maybe most importantly, on a really human level, it allows us to preserve fertility for people like cancer patients, giving them a shot at having a family in the future. It's incredible stuff. So all this leaves us with one big final question. We figured out this unbelievable trick. How to pause the life of a single cell, of sperm, of eggs, even of tiny embryos. We've learned to stop time on the smallest scales of life. So the question is, what comes next?